Okay. Then we can assess the overall proteins of cell and tissue also. See, each cell has a specific plethora of proteins which are expressed by the cell. So, the function of the cell depends on the collection of these various proteins which it expresses. So, that we can do by various methods like chromatography, there is ELISA, gel based approaches are there, spectrometry is there, X-ray crystallography is there. Then these days we also use bioinformatics analysis, which gives us the overall protein structure of various cells which are there. So, we will not go into details of each and every method, but that is the way we can assess the expression of proteins by cells and tissues. Then we can record the biopotentials, as we discussed earlier, that cells have an resting membrane potential and they respond to a stimulus by generating some potential change across their membrane, which is known as action potential. So, that is happening in neurons, in muscles. So, we can actually record the biopotential. So, there is ECG recording where we record the electrical activity of the heart. Then there is electroencephalography where we record the electrical activity from the cortex. Then electromyography where we record the electrical activity of the skeletal muscles. And then there is nerve conduction velocity, so by which we can actually determine that whether the conduction of the action potential along the axon is normal or not. So, here is an example showing the electrocardiogram, which is recorded by placing some electrodes on various areas on basically limbs of the body, and this records that how the electrical impulse is traveling through the heart, whether it is normal or not, whether the duration of travel is not normal or not, and the waves generated, is it okay or not, is there any abnormality. So, by analyzing this electrical activity, we can know a lot about the functioning of the heart. Then there is electroencephalography, which is the recording of cortical activity, and it is used in research for studying sleep physiology. So, what is the activity of the neurons during various stages of sleep, that we can identify by electroencephalography, and this also helps us in determining certain sleep disorders. So, in which type of sleep, that is in NREM sleep or REM sleep, we get some abnormal EEG activity, we can identify sleep disorders also. Secondly, it is also used to diagnose epilepsy, and from where it is actually generating, where is the epileptic focus. Then there is electromyography and nerve conduction velocity. Basically, in this we stimulate a nerve and then record the electrical activity from the muscle, and this helps us in identifying whether the duration of conduction of the nerve impulse from the nerve to the muscle, is it okay or not, and whether the electrical activity which is recorded from the muscle, is it normal or not. So, that will help us in determining the functioning of the skeletal muscle as well as the nerve. But here we are talking about motor nerve. Similarly, sensory nerve also can be analyzed, where we stimulate at one end of the nerve and record the action potential of the nerve from the other end of the nerve. So, that will help us in analyzing the functioning of the sensory nerve as well. Then finally, we can do cell culture. Cell culture is used for studying normal physiology and biochemistry. It also helps in assessing effect of drugs on cells or effect of toxic compounds on the cells, and also to study the nature of carcinogenic cells. So, what we do is we take out the sample of the cells and grow them in a culture where proper environment is maintained for the cells so that they can grow and divide, and then we can add certain toxic compounds on the cells and see that what is the effect on the growth and division of the cells. Similarly, we can take some abnormal cells or maybe infected cells and we can study the effect of drugs on cells. So, that is a method where we see that what is the effect of various environmental changes around the cells, what is the effect of these changes on the growth and development of the cells. So with that, we have discussed all these aspects which we considered in the beginning, that we can actually see the size and shape of cells, study gene function and mRNA, expression of the proteins. Then organelles functioning normally can be assessed by organelle specific assays. Then the proteins which are expressed, even the entire combination of proteins which are expressed can also be studied. Then electrical activities can be recorded from the various organs, and the secretions also can be studied by taking the sample and studying the concentration of various substances. Plus the damaged cells also, that when the cells are damaged, they release certain contents into the circulation. Thank you and have a nice day.